Hi, my name is Natasha Karp and I'm a biostatistician from AstraZeneca. I'd like to thank the organisers for inviting me to talk about sex bias in preclinical research, why it exists, and then explore how to drive change. First of all, I'd like to consider sex as a biological variable, explore the prevalence of sex having impacts on our experiments, then explore why a sex bias might exist in our practices, consider the sociological exploration of the topic, and then look at the challenges of changing practice. So let's start at the beginning. Sex as a biological variable. The neglect of sex within in vivo preclinical research is well established in the literature. There are numerous papers raising the issue throughout our research pipeline. For example, if we look in reporting, a paper in 2014 surveyed a number of manuscripts and found that 22% did not specify the sex of animals that they were using in their studies. If we look instead at experimental design, a paper in 2011 looked across 10 fields of biology and found that on average, 80% of the animals were male rodents. In 2015, Mazur and Jones raised the point that the sex bias had not changed in over a 20-year period, which means that the literature has been talking about it for over 20 years. And then if we focus on the analysis of data, even if we do collect both sexes, there's a tendency to discard that in the analysis and pull the data together in one bunch. And this isn't specific to the in vivo world, but also occurs in the in vitro studies. So again, we see around 70% plus of papers don't report the sex of the cells that they're studying. And in the design, it's predominantly male-only cells that are being used. And in fact, when you look at the provision of the cells, majority of cells are sold without defining the sex because it's not considered a relevant piece of information. So when I first started thinking and talking about sex as a biological variable, people said to me, but surely when it's female prevalent diseases, we would use female animals. And I thought, well, yeah, but okay, I'm going to look in the literature, see if anybody's researched that particular question. And that question was answered in 2014 in the review by Jon. And he looked at 45, he looked at papers published in surgical biomedical research between 2011 and 2012 for publications on female prevalent diseases, of which he had 45. 44% did not report the sex studied. And for those that reported the sex, only 12% studied females. So no, even if it's a female prevalent disease, it wouldn't necessarily follow that we would use female animals for the research. Now, we know from our own experiences that the prevalence of a disease, the symptoms we're going to experience, the progression of a disease, and the side effects that we might experience depend on our sex. And we can see that in a piece of research published in 2001 exploring the drugs that have been withdrawn from the US market between 1997 and 2000. And they found that the predominant reason was because of greater health risks for women compared to men. And these health risks weren't minor side effects. They were life-altering, disfiguring surgical complications, birth defects in babies, and onset of chronic diseases. And this we can see in the impact on the adverse drug reaction, in that a comparison between the males and females found a statistically significant difference with an odds ratio of 1.6 that women will experience an adverse drug reaction compared to men. Now we talk about personalizing medicine and we focus on genes, on genetics. Well, actually, we could just focus on the sex of a person and have a significant impact if we consider this in our pipeline. So in 2014, the NIH in the US, the National Institute of Health, took the unusual step that instead of recommending that we include sex as a biological variable, as so many funding bodies had before, they mandated it. They said, if you want funding from us, you're going to have to balance the sex in your cells and animal studies. So what did they ask? They said, we need to include females. You need to analyze by sex. And they coined the phrase sex as a biological variable. They also made this mandate for both cells and in vivo studies. And this is very relevant because the NIH are the largest funders of biomedical research in the world. So what was their expectations? Now, the thing to note here is that they're not asking us to understand how the treatment effect depends on sex. What they're asking us to do is to always include both sexes. So if there is a big difference, then you will be able to see that difference when you visualize and analyze the data, and it could set off a whole new path of research. And they rigorously defined exceptions, scenarios when you wouldn't need to study both sexes. So what are those exceptions? Okay, if you're working with pure molecular studies, such as how proteins interact, it doesn't matter the source material and whether you got them from males or females. If you're looking at sex-specific conditions, such as ovarian cancer, of course, sex is not a relevant variable. If you're working with acutely scarce resources, such as non-human primates, it'd be justifiable to study only one sex. And they did have to provide, of course, the other statement, if you can provide strong justification. But they were very explicit to say, absence of evidence regarding sex differences is not justification. If you have not seen a sex difference to date, that doesn't mean that you can just start studying one sex. That means you keep studying both sexes and estimating from them both the treatment effects. And if an effect occurs, you will be ready to see it. Now, the reason they're asking us to do this is, for example, because of this sort of issue here. And here I wanted to explore the impact of pooling our data together. So what I've done is I've artificially constructed some data that has a defined mean and variance, and I've added a sex effect for only one sex. And then on the left, I've just pooled that data together. And what you see after the treatment is that the variance looks a little bit higher, but not significantly so. That could easily happen in an experiment, and you wouldn't see a difference in the mean. But as soon as you visualize your data by sex, you immediately see, well, actually, that does depend on one of the sex, but not the others. So this is what they're pushing for. We need to visualize data by sex, and we need to statistically include a term to ask whether the effect depends on sex. Now, of course, this push from the NHS was greeted with a lot of controversy. The numerous papers raising issues with this proposal, and I particularly like the paper published in 2014 by Professor Field, who stated it was a major step in the wrong direction. It was a waste of resources, and we were scientists and we should be trusted to know what we're doing. But if we think of the work by Johnson in PLOS One 2014, what they raised is there's a knowledge gap. If we've always been studying one sex, then how would we know whether this variable depended on sex? We sort of just got a pattern of behavior that we wouldn't know. We have a knowledge gap. And at this point in time, I was working with a large data set that actually had both sexes. And I thought, aha, this is something that I could actually answer with this data set. So let's explore that. So assessing the prevalence of sexual dimorphism. So I was working with the International Mouse Phenotyping Consortium, which is a group from all over the world working together to knock out every single mouse coding, protein coding gene in the genome, and to then characterize the resulting mouse line with a broad set of tests so that we could relate the genotype to the phenotype. And for each knockout line you were making, you would generate 7 females and 7 male knockout lines. And we generated both because we were breeding the animals in-house. And so we studied both because otherwise we'd just be throwing away one of the sexes. And what you can see on that graph on the right, the diagram on the right, is the sort of tests that were happening for those animals. So for example, at week 9, you would be conducting tests such as open field, to look at their behavior when they run around a box, or grip strength, whilst at week 14, you're looking at body composition, and week 16, you've got variables such as heart rate or the clinical blood chemistry parameters. So it's a broad spectrum of biology is being explored. And this work was, that I'm going to present now, was published in 2017 in Nature Communication. And to give you a sense of the scale of the data that I was working with, I had data from 14,000 different wild-type mice, 40,000 different knockout mice that looked across over 2,000 different knockout lines, looking up to 234 traits. And so the idea here is I'm not asking for a particular treatment, does sex have a role in explaining the effect, but rather, if we look at a whole, if we were running any sort of experiments, how often would sex have a role? Because this is just a representative sample of people doing experiments. So there's 2 ways we can ask this question in the data, the role of sex as a biological variable. We can first ask, how often does it change the behavior of the data in the control data, such that if you pooled the data and didn't include those terms in the analysis, you would be making mistakes and you could be having causality issues in interpreting your data. So we have two types of variables in this pipeline. We have continuous variables such as lean mass or sodium level. And we have categorical variables such as the rib shape is abnormal compared to normal. So if we look at the categorical variables, which is normal versus abnormal, we were in the pipeline looking for rare events where the knockout of the gene would lead to an increase in the abnormality rate. But we were surprised to find that even in the control data, there was a difference in the abnormality rate between the males and females for 10% of the times. And sometimes the abnormality rate was higher in the males and sometimes in the females. If we think instead about the continuous variables, such as the sodium level or the body weight, again, we see a difference between males and females, but for 60% of the variables. Sometimes the mean was running higher in one than the other. But maybe more interesting to ask, how often did sex modify the treatment effect? And here we've got two pie graphs, one for categorical and one for continuous. And this is a pie graph of when we first detected a statistically significant effect of knocking out the gene, we then asked, did it depend on the sex? So this is showing of the ones that were statistically significant, did it depend on the sex, the effect? And we find for the categorical that 13% of the time it did. Sometimes the abnormality rate was higher for the knockout in the males, and sometimes it was higher in the females. And with the continuous variables, it was around 15%. And this time it wasn't more that it was in one sex only per se, it was more that it could be a different size between the two. Now, we only had 7 males and 7 females for each knockout gene. So we weren't particularly powered to see a sexual dimorphism in the treatment effect. So this indicates that the effect is quite prevalent. And so sexual dimorphism is interacting with our treatment effects at a high proportion of our experiments. So then why does a sex bias exist in our practice and how we run our experiments? And I would argue that this arises from some misconceptions, some skill gaps, practical concerns, and the 3R interpretation. I'm going to focus on the 3R interpretation because for animal experiments, that's actually one of the major players that's playing out with how we conduct our experiments. When we run experiments, we have to isolate cause and effect by simplification. We have to choose where we're going to simplify, and we simplify down to a standardized space where the only difference is the treatment. And there's always a decision point of where we do that. Because of the 3R ethical framework, we also need to simplify so that we can draw decisions, but we've got a lot of pressure to minimize the number of animals used per experiments. So under the 3Rs and the reduction element of our ethical decision-making process, the historic interpretation of reduction was to minimize the number of animals used per experiment. And you would see conversations in the ethical review boards discussing whether to use 7 or 5 animals per group in experiment, and a lot of energy focused on this. Now so if you can cut down from 7 and 5, that's considered a 3R benefit and something that is highly valued. Yet it doesn't count the fact that if you've used 5 males here, there have been females bred in that process that are then being discarded because they don't get counted in your number. It's the numbers you use within your experiment. And the fact that you can't actually answer the question for the females isn't considered relevant either, because you're focused within this experiment using as few animals as possible. And if you only focus on one sex, people perceive that they could use less animals. Now, there is now a change in the definition of reduction, but it hasn't actually become truly embedded in our culture. And this was proposed by the NC3Rs to appropriately design and analyze animal experiments that are robust and reproducible and truly add to the knowledge base. It doesn't talk about N anymore. It talks about adding value in the experiments, being sure that the experiment is valuable and therefore justifiable because the data is going to add to the knowledge base. So there's often a question, well, why did they choose male animals and not female animals? If we were going to just choose one sex to have a simpler testing space and reduce the number, why did it always end up being males? And that's because of the misconception that female mice would be hormonal. And because of this hormonal variability in the mice, they could be at different stages of their cycle. They would be more variable. Now, this is actually a wives' tale. The piece of research published in 2014 was a meta-analysis looking at 293 published articles where they had studied both sexes. And it compared the variance between the males and the females that were not, and were at any point in their cycle, of their estrogen cycle. So they were random stages. And what they found is that when they did a global analysis, and this is shown in diagram A on the top on the right, there was no statistically significant difference between the variance seen of the female and the males. And if we instead look at individual variables, we do find 3 variables that are significantly different, but that's because the variance is higher in the males, not the females. So this is a sort of very embedded cultural concept that females are more variable, but isn't actually evidence-based. Females are no more variable than males, and that shouldn't be used as a justification that you should only focus on males in your research. The other reason that I think people focus on one sex is that if you study one sex, you have a control group and a treated group. You can explore the data. It's very simple to visualize. It's very simple to analyze. So you're basically we're looking at one sex, you've got for a continuous variable, a student t-test. You're looking at the variation of the variable of interest, y, it depends on the treatment. You can do this in Excel. It's very easy. However, if you move to both sexes, you're now doing a two-way ANOVA. You're asking, what is the effect of treatment after accounting for any sex differences? And does that treatment effect depend on sex? Can't do this in Excel. You now need to use statistical software. And in reality for our biological communities is that we've had very limited training in our degrees and PhDs in experimental design and statistical analysis. And we can see that in that statements like 4 out of 10 Americans will publicly say that they hate mathematics. Mathematics anxiety is recognized as a physiological condition. And if for each time you add an equation to your manuscript, you will get 28% fewer citations. It is socially acceptable for a biologist to say that they hate statistics and mathematics, yet we actually need it for our research and our everyday studies. So I perceive that this is a skill gap that is playing out, inhibiting people from embracing these more complex designs. One of the other issues I would like to raise is the misconception that it will double my animal usage. If I say to someone, okay, but why don't you run the experiment with both sexes? They say, but that would be double the number and I just can't afford it. But the actual advice is to keep doing what you're already doing, but change half the animals in your study to females. And people go, but how can you do that? Your statistical power won't be high enough. And that's because you're moving from a randomized complete design to a factorial design. When you're working in the diagram on the left, which is for a single sex, you're estimating your treatment effect between the control group and the treated group alone. But when you're working in a factorial design, you'll be estimating your treatment effect after accounting for the sex differences from both the males and the females. And that is why the impact on power is not as significant as you would initially expect. Now, there are a lot of the things there that we're saying that are misconceptions or they're skills gaps, but actually there is some issues with stepping up your experiments from studying one sex to 2 sexes. You have increased the complexity of your experiment, particularly when the fact that we're dealing with living creatures. To manage the welfare of our animals, say rodents, we cage them by their own sex so that they're not lonely, which means if you're going to try and reduce order effects with time, you can't keep going in and out of cages because 1, you're going to make mistakes, and 2, you're going to distress the animals. So you have to process them by cage. But how do you minimize order effects? How do you manage the fact that the pheromones of the female animals might impact the male animals? Should you clean the equipment between the sexes, or should you make a scent cloud? So there are lots of issues and practical concerns. Will the exposure to the female animals increase fighting between the males, which becomes then a welfare concern? Now, the solutions to this are going to be very dependent on your environment. Do you have closed cages? Do you have open cages? What exact tests are you running? But people out there are running experiments with both sexes. So we need to find out practically on the ground exactly how they're doing it. And that is information we need to share because we need to work out the right decision to balance how to run these experiments practically with both sexes, but manage the welfare of the animals appropriately. Now, one of the things I wanted to touch upon is that in the beginning I said the NIH was requesting this change to both sexes for both in vivo and in vitro. And that's because we can see within the in vitro community that there's a similar bias, the use of male cells. So what's driving that behavior in our research pipeline? And I believe that's a misconception, that the sex of the cell is irrelevant. And that usually when you raise, some evidence, people go, oh yeah, of course, I hadn't thought of that. And that's because people believe that once you take the cell away from a hormonal environment, that it doesn't matter whether it's male or females. But now with our thoughts about epigenetics and gene expression, we do, there's much more evidence to suggest that it's not just the hormones driving the difference, it's at a genetic level. And there was a piece of research that I published in 2007, which was a really nice example of demonstrating how sex matters. And this was looking at stem cells treatment for muscular dystrophy in an animal model. And they were having real variability in their response. And they could have just walked away from the experiment, but instead they decided to explore where the variability occurred, and they ran a factorial design. And what they found was that the ability to trigger regeneration depended both on the sex of the donor and the sex of the recipient, and that the female cells were much more robust to the process, leading to a better outcome than the male cells. So this insight opens the door to further opportunities for research, but if they hadn't thought about sex as a biological variable, they would have easily missed this. So, we've reviewed the sex bias in our pipelines and seen that it's well established both in vivo and in vitro. So what we need to now consider is the challenge of change. Why is it we have manuscripts that have been talking about these issues for many years with no progress and no change in the practice in our labs around the world? And the work that I'm going to talk about here has been written up in an article in the British Journal of Pharmacology. So I'm going to explore a number of management techniques to help us understand why change is difficult or what we can do to drive that change. And one of the things I want to start with is the culture. So the culture is very much how we do things around here. And a useful model to understand culture is the onion model. And in an onion model, you have the outer layer of the onion, which directs our behavior, which is the formal guidelines, officially what we state we're going to do. This is then implemented into actual processes and workflows of how we're going to do things. But really what drives our decision-making process is our underlying assumptions. Now, these are unconscious beliefs. These are things we are unaware of that are driving the decisions. And the concept behind the onion model is that it's easier to change the outer layers of the onion than the inner layers of the onion. But if you actually want to change practice, you have to change the heart and mind and tackle those unconscious beliefs. And if I have a think about what I think is driving our decision-making process, it's not possible to include both sexes. I couldn't justify it from a 3R perspective. It's going to double the number of animals, and there's no benefit to including women. We've done all this research to date with just one sex, and it has delivered. There isn't going to be a difference here. And these underlying beliefs are the ones that are triggering the failure to make change to our practice. So what's useful then to think about is that when you're trying to make change for an individual, how do they emotionally react to the change that you're trying to deliver? And so this is Kerber-Ross change curve that was originally developed for the concept of grief, and it has since been applied by businesses to business changes that are needed. So, you have your initial reaction of shock or denial and anger, where you then try to bargain the issue away, which can lead to depression, and hopefully you move forward to acceptance. Now, if we relate that more to a business scenario, what we're hoping to see as people start to embrace the change is that they start to experiment with the idea, and then they'll make a decision whether to be open to the idea or not be open to the idea, and hopefully they'll integrate that into their practice. And the thing to note about the change curve is that people move through this curve at a different rate. And also that the initial reaction when you ask people or suggest an idea to them is shock, denial, and anger, basically resistance. And so you shouldn't take it personally. You need to ride that emotional reaction and see it for what it is. It's fear of the change that you're proposing, and that you need to keep revisiting the idea so that you can drive that change forward. And the other thing to note that I think is useful is that if you've been working this maybe as a manager for a while, that you will have traveled this curve and been coming out the other end. When you present this to your team or other people, they're going to be back at the beginning. So you have to accept that people will be at different points in this curve. So what we need to look for are strategies that help people move along this curve. So Gary Yoke has written numerous books talking about influence tactics, and he says that these strategies can have three reactions. They can lead to commitment, reluctance, compliance, or resistance. So resistance is actively digging your heels in, undermining what people are suggesting, saying that's a stupid idea because of this, recruiting others to the resistant cause. Reluctance, compliance is that when people will do it when you're looking at them or you're sat with them. But commitment is what we really need, which is people become advocates for the idea that you're proposing. So what strategies could we use? And well, here are some of them. And Gary grouped them into two types: positive strategies and negative strategies. Positive strategies led to commitment, and negative strategies led to resistance. So the positive strategies that he suggested were things such as rational persuasion, such as this talk, debating and discussing this very idea to try and win the hearts and minds of you that we should be considering sex as a biological variable. We could have exchange, which in a way is the NIH have done. They're not going to try and win your hearts and minds, but if you want their funding, you're going to need to have to do it. There's a consultation process. A consultation is another strategy because you start to unpick why people aren't embracing sex as a biological variable. Is it there's a skill gap? Is it they don't know how to manage the welfare issue? Is it a misconception that it's going to double their animal numbers? We can also do an inspirational appeal. We can highlight examples where this practice is run, such as in the Jackson lab. Most of their behavioral studies are run with both sexes, and that is considered a world-leading institute. Now, negative strategies are pressure. You will do this, and then people just dig in their heels. They just hit with the anger, and they don't move any further forward on that cycle of embracing that change. Now, there's two strategies that Gary put in negative. So legitimizing. Now, legitimizing to me is a little bit like inspirational appeals. If I say the Jackson lab are doing it, it can come across as that I'm saying they're doing it, so you should be doing it. So it comes across as a pressure strategy. So it depends on whether you can do it as inspiration or as a pressure approach. And coalition building can be a really valuable resource in driving change, but it depends on how the coalition work. If they then just jump to providing pressure, it isn't going to be successful. If instead the coalition work to do rational persuasion, inspiration, and consultation, then it will be successful within an organization. So this tool here is a force field analysis. And what you do is you quantify the forces driving the change and quantify the forces resisting the change on a qualitative scale of 1 to 4. And if the forces resisting change are stronger than the forces driving change, there will be no change. The status quo will be maintained. So if you want to drive change, you have to strengthen the driving forces and you have to weaken the resisting forces so that you can defrost the current status quo to refreeze in a new situation. So if we look at the forces driving change, well, there's women's health. Now that is gaining power at the moment. Women are gaining a voice. So that is becoming a more significant force. There's the replicability crisis, again, something that's gaining power. Journals have an influence, funders. And then I've put in light gray. These are groups that can have a lot of influence, but it depends on your institute. There could be ethical boards could be quite powerful at driving this change. The institute culture and how they approach it will set the standard of what is normal within your institute. And the leadership, the management leadership, either within your professional bodies or within your institute, can have a lot of impact on this. But you can see from the dark green that the actual forces driving change are quite small, which would explain why the status quo has been maintained. What's resisting change is very much our culture. The 3R interpretation that we have to use as minimal animals as possible, and we can only do this through using one sex because we don't quite understand how power is calculated or how to use a two-way anyway. There's not how we do it around here. I've always done it this way, and so there's lots of resistance arising from that. There are some concerns about money, and whilst you might not be doubling the number of animals, you may well be using animals in different cages that could play out. There is an increase in analysis complexity and practicalities, and there's a belief in the values. but most of these are cultural and the misconceptions. So these forces can be weakened if we focus our minds on that. So with that in mind, what we need is an institute level plan. How can we drive change within an institute? And Professor John Cotter, who from Harvard, has written numerous books on this. And one of the 1995, he put forward this 8-step change process for leading change. And what I like about it is that he talks about how all the early steps are about leadership and the later steps are about management, because you have to have the leadership steps so that you win the hearts and minds and change that inner core of the onion model. And instead of jumping, what we normally do is we jump to how are we going to practically change these activities on the ground and what work streams are we going to change, which is the focus on the management stages. And that's not going to work. He talks about how you have to work through the stages sequentially, though some of them will start to be running simultaneously. And he also highlights that you have to get all the way through to the end, or any progress you make will be lost and you will revert back to historic behavior. So the first steps are around creating urgency, a burning platform, which the NIH is doing very well for us, that, you know, if you want their money, you need to embrace sex as a biological variable. And then suggest forming a powerful coalition to then do the later stageships to provide that leadership, using people all across the organization. People have influence in the animal houses, who are key players in the therapy areas, who are key players in the ethical decisions. They're the people that can drive this change. But you will need the figurehead as well from the professional bodies of the institutes. You need to create a vision for change, and that vision needs to be a soundbite. It needs to be something that you can quickly understand and get your hearts in. It can't be a 4-page document that nobody's really going to read and is going to be signed and put in online. And the reason it's got to be the soundbite is you have to communicate the vision with high repetition. And in the books, he talks about how much information we receive in a typical 2-month process. So if someone says there's a new initiative, does a talk, does a poster, initiates a presentation, that's just going to go, and that's very interesting, and it drifts away to nothing. If we really want this to change, it has to be something that we're talking about numerously, frequently, and has been raised multiple times in this area for it truly to have an impact. And only then, when we've done those steps, can we start to think about removing the obstacles, removing the blockers that prevent people from doing this, so we can create short-term wins. When you create the short-term wins, we're helping to maintain the energy to keep going forward, to help communicate the vision and to keep everyone that sense of urgency going. And you can use this to refine your vision and to refine the communication going forward. And only with these cycles will you anchor the change in the culture so that it truly embeds as a new practice. So that's a sort of a business management perspective on it. But I also had the wonderful opportunity of reading a sociological exploration of the issue, because I got to read Annika Gomper's master's dissertation at Cambridge University. And she interviewed 9 different scientists in the US to ask them what did they think about sex as a biological variable in their research. And sociological exploration is qualitative research looking for patterns of meaning or themes. And she identified 3 different themes and said that they were conflicting. So the 3 themes were generalizability, avoiding complexity, and practicality. She highlighted how all the scientists completely believed that it was really important to research, to understand variation, to understand the biological story and the difference. What is happening requires us to explore variation. So it was really important to consider sex as a biological variable. But we need to avoid complexity. To make progress in science, we reduce complexity. We have to simplify all this variation that could be influencing the story into a simple testing space, so we can make a decision and communicate our finding and incrementally step forward the science. And so there are practicality concerns. And she's not talking about the practicality of running an experiment. She's talking about the practicality of the scientific process, because there's a mediated tension between the generalizability of our conclusions. We want to make a general conclusion about what we found and avoiding complexity. If we want to make a conclusion, we can't say, well, it depends on this age and this sex and this diet, because we sort of get lost amongst the trees. We're trying to convert the question into a doable problem. So we are actively avoiding looking for a sex difference because we want a simple answer. We need some examples where people have actually done this. and, and show us a way forward. And through that, it will become much easier for us. So, in conclusion, sex bias is culturally embedded in our research pipelines. It is impacting the reporting, the design, and the analysis. Now, precision medicine is touted as the future, and we typically focus on differences in genetic factors, environment, or lifestyle. But I would argue considering the sex of the patient provides an easy win to optimize the healthcare. So that should be our first port of call. And researchers suggest that sex is a significant source of variation for both in vivo and in vitro. So whilst we've not before looked, that doesn't mean that it isn't playing a role in the story. This isn't an individual science issue, but a scientific practice issue. We need, as a community, to actively be discussing these issues to support scientists to be more mindful of sex as a biological variable in our pipelines. I'd like to acknowledge numerous people who've worked with me over the years who have helped me along with this story, particularly the IMPC and the statisticians in Tel Aviv University in Israel. Thank you very much for listening. I hope you enjoyed the talk.